they never sprayed any pesticides. 2008, suddenly the entire potato completely burned within 3-4 days. We have around 50,000 hectares area was there, one district, entirely complete damaged. There was a issue in Karnataka Assembly also. Then later on, Bangalore surrounding areas, within a matter of 1 week, most of the tomato planters got dried up. Then we entered the picture and find out how this happened. This is not required, I think. So here, see, when you look at the global scenario, so the entire, all the either complete all the potato growing areas, there was only one species, one group of Phytophthora infestations was there is PAM1 genotype. In all the 6 continents, this is the only one Phytophthora host specialized group was there. At the time, not much epidemics were there. Later on, when the population started changing, when you look into the UK, there is a clonal population called 13A2 genoclonal population. This 13A2 clonal population arose somewhere in the 2006, and all the resistant cultivars like sterling potato also got broke down. So at the time, then started analyzing the population. The same thing happened in US also. It was the PAM population. Later on, it was changed to the US1. But finally, that population is US22. That became aggressive in entire USA. Started epidemics, and all the tolerant birds are broken down. So when you seen the genome sequence of Phytophthora infestations, it has got a 230 MB GB. Yes, 230 MB. Sorry, 230 MB. At the same time here, it has got some sort of zinc region more, and it is able to produce the effectors, particularly RXLR effectors. So here, I would like to emphasize, when epidemics of Phytophthora late blight occurred on tomato in 2006 and 7, when analyzed the population by using the different molecular markers, it may be RFLP, RG57, mitochondrial DNA haplotypes, and metalaxyl resistance SSR markers. All the isolates we recovered from the tomato or metalaxyl tolerant. They found to be the population called 13A2 clonal population, which is not occurring in India before that. Because of this 13A2 clonal population emerged in India, that's how the epidemics have occurred, started creating a destruction of the potato and tomato, both in Karnataka. Later on, it moved to the northern part of India. Now, this population is also available in West Bengal also. This we published. So, when you analyzed where this population has come from, so if you look at the potato imports from other countries to India, so where potato import from Netherlands 2005 and 6, some company to Karnataka, they started giving to the farmers for what you call contract farming for chips production. So along that potato tubers, the new population came from the Netherlands to Karnataka first. Karnataka then started a destruction of potato crop in Asan district of Karnataka. From there it moved to the entire Karnataka and destructing both tomato and potato. That's what happened. So if you look at the conditions in Europe and India, so though the conditions are very low in Europe, but our conditions are even more than 25, that population when entered to India, it has adapted to the local conditions and started causing epidemics. So these are the parameters what we have analyzed. I don't want to go details. And we developed some sort of computer-based model also for forecasting. We found that August 11th, 2008 to even today also, August 11th to 15th, the disease definitely will start in Karnataka. So we are advising farmers, before that you can give the what you call preventive sprays. It is very clear, every year. August 9th to 15th, disease will start invariably. So this system is working very well. And then we started giving different sorts of fungicides, and finally we developed a complete package for the tomato crop, and removal of the stacking, removal of the leaves up to 30 centimeter height, putting the plastic mulch, and giving the Bordeaux mixture first, the first spray. Even it was not there practice in vegetable crops, though we are practicing very well in plantation crops. And you give the Bordeaux mixture, I think the disease veterinary can manage well. You can look at, see here, the control and uncontrolled one. This is one example. So how the actually new population came to India from Europe and triggered the epidemics, caused the losses to the farmers. The second one, chili. I wanted to give one more example, chili. Chili, we have a Phytophthora capsicum causing a disease here and there. But 2011 and 12, we have seen the lot of epidemics of chilies causing Phytophthora, either leaf blights or stem rot, root rot. You can see the entire plantations are drying up. Even the company's material also lost many people there. So when analyze the population here, see, to our surprise, earlier Phytophthora capsicum has been reported causing the blights in chilies, and we found that a new species called Phytophthora bohemia is a homothallic species, which was reported on cotton in China a few years back. It is not a problem in India, and this has become a serious epidemics. And you look at this aggressiveness, see the Phytophthora capsicum isolates are first lines, you see, they are aggressively low, but Phytophthora bohemia has become very aggressive. Because of this Phytophthora bohemia replaced the old population of Phytophthora capsicum and caused the epidemics. This is published in Plant Disease. And one more example, like actually, where we have seen Phytophthora nicotianae occurring diseases on tomato, brinjal, and ridge gourd also. So when we isolated, we are surprised. Nicotiana is not new species in India. It was occurring causing the buckeye rot in tomato, but we never seen much on brinjal, and no reports on ridge gourd. So we wondered what is this Nicotiana causing here. Then we started using the specific primers and identified Phytophthora nicotianae. Then we started analyzing the RFLP patterns of ITS. All the isolates from either brinjal or tomato or ridge gourd looks uniform, no difference are there. You can see even we used the protein profiles, all are three same, and even used the isoenzymes, no difference are there. Then we started a networking of mitochondrial genes, but all are looks alike only. Then we went for a nuclear genes, and we could able to differentiate very clearly. Tomato group is separate, ridge gourd group is separate, and brinjal group is separate. So based upon this, then we started doing what we call assay, infection assay. We could able to see tomato isolates are highly infective on tomato, brinjal isolates are highly specific on brinjal. We could able to differentiate very clearly. Here I would like to recall Dr. Subbarao. So 2016, this paper was submitted to phytopathology. When he came to Delhi, I spoke to him. So it was published in the phytopathology.
You didn't publish because I said so.
No.
The paper was good.
Just mentioning you, no. So, it has got merits, no issue on that. But here I would like to mention, we have seen the three groups here. One is the Phytophthora infestations population coming from the Europe and triggered epidemics in India. The second one actually Phytophthora bohemia, which was not there in India and causing epidemics on chilies. And third one, the Phytophthora nicotianae adopting different crops, producing different diseases. So based upon this experience and have a complete data, I would like to mention there is a strong biosecurity required in India, either domestic or international also. So beside this, we have seen the lot of problem in coconut also. We never had I will stop here only, further I will not go because I have a lot of slides are there. Time also is limited because further I have so many crops are there. I will stop here only. So before stopping, I would like to mention one point only. See what happened, coconut. We never had a mite on the coconut. 1998 came to India. Within a matter of few years, the entire coconut areas completely created a havoc. And one of the ministers in Karnataka lost minis also because of this disease, this pest. Then later on, slowly it is coming down. Naturally, it has managed now. So 2016 and 17, there was one more pest called a spiraling whitefly. The spiraling whitefly, we never had problem. Somebody brought one ornamental palm from Florida to Pollachi in Tamil Nadu. Somebody brought. We know who has brought also, just like potato who has brought from the Netherlands to India. I don't want to tell their names here. When he brought there, small patch is developed there. Within a matter of 2-3 months, it spreads the entire Kerala, entire Karnataka and Tamil Nadu, Andhra Pradesh. Now wherever coconut is there, you can see the black coconut. Coconut is always green and smiling. If you look at now, most of the coconuts, when you just travel, you see, most of the coconuts become black. Because of spiraling whitefly, comes on the underside the leaf, produce secretory nectar. Correspondingly, you get the sooty mold. Coconut become black. So many people, farmers are crying. No control method is there. And spraying one other issue. And we suggested drones also. And drone also will not spray properly. Because even I used the drone recently. I have big mango. I am no longer scientist now. I am a farmer now. I grow ultra density mango now. So I used the drone recently for a spraying. I did not put much happy on that. Then we went for manual spraying. These are the issues are there still. I don't want to go much. This all. This is blackbird. See coconut fight after. Thank you very much. I don't want to take much time. So I.
Kindly give big hand to Chandra. And it's a very diversified infection with different crops. I think I will request Dr. Subbarao to kindly honor Chodappa by giving memento, shawl and a certificate. You can go on the dance. Very good. Thank you. Thank you, Dr. So now we have two plenary lectures. One of them is by Dr. K.S. Subbarao himself, Beyond the Lab, a journey through discovery and innovations. And the second lecture will be by Dr. P.K. Chakrabarti, former member of ASRB. He has not specified any topic. इसमें तो नहीं है enabling. Enabling innovations, aligning agriculture policy with field realities. Okay. So we have 20 minutes.
While they load,
Yeah.
let me say a few words. It's a pleasure to be back here. I enjoy coming to IPS meetings each year. I have been doing that since 2016, except during a small interregnum during COVID. So thoroughly enjoy the visit.
You can speak to the mic.
Yeah. What I did, unlike previous times when I focused on a single topic, what I have planned for you is to give you a snapshot of my entire career in about 30 minutes. I prepared for my talk for 30 to 35 minutes, and I was about to leave for the airport. That's when the program came, and it was reduced to 20 minutes. And there was no way I could revisit the talk and collapse it to 20 minutes. So if you ask me to stop, I'll stop. If not, I'll complete it. So What I did was I gave the title simply because you will see my journey from a young researcher to where I reached in the end. You will see a few of these projects that have provided me immense satisfaction as a plant pathologist. So I want to highlight a few of those for your benefit, and it's a pleasure to do so. Where did it all begin for me? I grew up in a town very close to the city of Mysore called Periyapatna. I got all my college education in Mysore, including my master's, and I secured a CSR fellowship to continue on to my PhD. So the project was on fungi, but it was not on plants, it was on human beings. As you well know, fungi also cause many diseases in human beings, but they are secondary in nature. They only occur in immunocompromised patients. So at that time, the doctors were unable to recognize or diagnose the fungal infections, separate them from bacterial infections, and they continued to treat the patients to bacterial infections, but the patients were deteriorating. So my project was to develop these diagnostic methods to identify the fungal infections in human beings. It was a very interesting project, and the project was going well. I got my first paper out of the preliminary work that I did, but the family situation was that I had to go get a job. So being the eldest, you know what the situation is. So it was incumbent upon me to support the family, so I went and found a job at XXIII as a research associate. This allowed me to reconnect with plant pathology, and it wrote the rest of the chapters in my long career. At XXIII, I was asked to work on a disease of pearl millet called pearl millet smut. As you know, pearl millet is one of the cereals that has the highest protein content in it, and it's a staple in India and Africa to millions of people. So pearl millet suffers from two panicle diseases called smut and ergot, and both of them convert the grain tissue into either sori or sclerotia. And for this reason, E.S. Luttrell from University of Georgia called them as tissue replacement diseases. So my project was to develop a screening method, and there was a very comprehensive paper written by Dr. R.S. Bhatt. It was almost complete study of this particular disease. And I wondered, what can I do new? He has already worked out all of it. It was a single paper. And yet, in the discussion, there was one sentence that caught my eye, and I'll explain to you what that is. Pearl millet is a protogynous crop. That means the gynoecium, style, and stigma come out of the florets first, and that remains fresh for about 7 to 10 days, and then the anthesis begins that you see on the right-hand side. Anthesis begins from the top, and that is in a basipetal fashion that this happens. So, my supervisor had worked out the precise details of the flowering events, and what I did was to select a few of these areas to inoculate and find which stage provides the best infection. And I found out that it is in the boot stage that the infection is maximum. If you inoculate the boot stage with the smut fungus, then the infection is maximum. But what I noticed, true to what Dr. Bhatt had said, when you inoculate, when anthesis occurs, the disease no longer develops. So the question is why? So what happens is, when in the protogynous stage, if you pollinate, if you provide the pollen grain germinates and fertilizes the ovary within about 12 hours. And the pathogen that you inoculate at the boot stage takes about 15 days for symptom expression. For it to reach the ovary, it takes about 10 days. So in the meantime, if you pollinate the florets, what happens is there develops a post-pollination constriction in the style and it collapses the stigma altogether. Even if the pathogen is there in the various stages of development in the style and stigma, it will be completely destroyed because it withers away. And this was the observation that we made in the greenhouse and laboratory studies. So how can we exploit this one for practical disease control? So as you know, pearl millet has a lot of F1 hybrids generated through male sterile lines, and they're all high yielding, and yet they're highly susceptible. So what we did was to identify a pollen donor line whose antithesis matched with the protogonous stage of the high-yielding hybrid. So we planted this in various combinations, either every 5th row or every 9th row, or mix it in various combinations and plant it. And if by simply offering this pollen donor at the right time for this high-yielding hybrid, you eliminate the disease altogether. So we deployed this in the growers fields, and I understand about a couple of years ago, a colleague of mine said this method of control is still being used by the growers, and nearly 99.9% of all the disease can be eliminated, both ergot and smut, by just providing the pollen at the right time. While I was busy doing these things, a professor from Penn State University, Dave McKenzie, visited XXIII with 4 of his students and gave a rousing seminar. So I was so fascinated with his seminar, I wrote to him at his Penn State address because I wanted to do my PhD with him. I wrote to him at his Penn State address, which is here. Here. But his response to me came from Louisiana State, and that's how I ended up in Louisiana. I arrived in Louisiana not having seen a wheat plant in my life, and yet my PhD project was supposed to be on leaf rust of wheat. As you well know, leaf rust was the most researched topic until that time. So what is new? How can you find something new to get your PhD work done? I spent about 6 months in the library scanning every literature there is. And what I learned was, it is the top 4 leaves of wheat that are responsible for the final yield. That is what the literature said. But what I wanted to find out is if it is true. So I retested that one. I created this elaborate treatment structure where all the leaves were defoliated or all the leaves were retained, and in between various numbers of leaves were defoliated or retained. Since the green tiller itself also contributes to photosynthesis and the final yield, all of them had the green tiller there. That's what I call the non-foliar parts. And I measured various yield components in wheat and developed this model that you see here. So what this model denotes is the intercept denotes the contribution of the non-foliar parts to the final yield, and each of the coefficients that are associated with the flag -1 tells the relative weight of the contributions of each of these leaves. So from this model, I showed it is not the top 4 leaves that are responsible, but it is the top 3 leaves that contribute to 99.9% of all the yield. So I submitted this manuscript, and many of you have heard of Larry Madden, who is probably the foremost epidemiologist of our time, and he was the editor that handled that manuscript. I was nervously waiting for about 4 or 5 weeks, and then one sentence in his decision letter really surprised me and elated me as a student. He said, "This is perhaps the most original contribution to crop loss assessment in the past 33 decades, is what he said." And you can see as a student, getting such a line from him was such an elevating feature for me. So then what happened, I continued at LSU and got some postdoctoral experience working on Soilborne diseases. And then I moved to University of California, Berkeley for another short-term postdoc. And then I joined University of California, Davis as a faculty member. So unlike a new faculty member where you have to search for projects to continue to work on, the growers in that area had already decided these are the two diseases that I will work on. So when I landed, I had already the research projects available and also the funding. They were willing to fund all of that. So I didn't have to struggle for funding either. So I'm going to talk about two projects on the Sclerotinia species. Both of them cause the same disease on lettuce. One is a strictly soil-borne pathogen, this Sclerotinia minor, and Sclerotinia sclerotium causes the same disease but through the aerial mode. So it is a deadly disease. Nearly 15 to 20% of all the lettuce that is produced can be gone, whereas Sclerotinia sclerotium, because it causes the disease through ascospores, airborne ascospores, the devastation can be 100%. So I will talk about one project from each of these pathosystems. So as I told you, Sclerotinia minor is a strictly soil-borne pathogen. So the sclerotia accumulate in the upper soil profiles. To remove the sclerotia from the upper soil profiles, they develop these massive plows so that you can invert the soil to a depth of about 60 centimeters and then remove the sclerotia that are located in the top profiles to deeper soil profiles where they don't cause the disease. But intuitively, it all sounds very good, but nobody had done the research. But the growers were already using this. These massive plows were developed, millions of dollars were spent, and they were continuing to deep plow in these things. The first project that I took after I moved to UC Davis was this one, to assess whether it actually works. So what we did was to sample the soil. When you deep plow, what happens is it leaves massive clots of soil. So to break these soil clots, you have to use double the number of tillage treatments than you normally do. As you can see here, in a conventional tillage, you have 11 passes of the different tractor operations, but in a deep plowed field, you need 22 passes of the same operations. So this massive tillage operations, dealing with the soil-borne pathogen, I knew it had to have some effect on the distribution of the inoculum that you took to deeper profiles. So that's what I did, was to collect the sclerotia before deep plowing, immediately after deep plowing, and after one crop after deep plowing. And what you see here is a statistic called Lloyd's Index of Patchiness that tells you the aggregation of the inoculum prior to deep plowing, immediately after deep plowing, one crop after deep plowing. So prior to deep plowing, the inoculum was highly concentrated in a few spots in the field. But with these increased tillage treatments, what you did was to drag the inoculum to every part of the field. And as you can see here, one lettuce crop after deep plowing, the inoculum distribution was nearly uniform. And that's how the disease incidence also was extremely high after deep plowing. So we showed clearly that deep plowing doesn't work. And one of the first lessons that I learned was that it is not only important to develop new disease management methods, but also eliminate the methods that no longer work. So as you can see, within 2 years after we showed that it doesn't work, the growers stopped using it. Just imagine the millions of gallons of diesel that have been saved because they no longer work, this deep plowing that doesn't even work. So it was a very satisfying project for me, so I just wanted to highlight that one. And onto the second project with the Sclerotinia sclerotium. It's an ascomycete, and you see the beautiful ascus with 8 ascospores located in the far right-hand side there. And in ascomycetes, you have both heterothallics and homothallics, as you well know. So in the homothallic species, what you have is the mat locus is located within the same individual. Whereas in the outcrossing, this heterothallic species, the mat genes are located in different individuals. They both have to come together for sexual reproduction to happen. So I will show you a very interesting phenomenon that we discovered in Sclerotinia sclerotium. So Sclerotinia sclerotium is a homothalic species. Both the mat genes are located within itself. So what happens in some ascospores is that the MAT1, portion of the MAT1 is actually attached to the MAT2 gene here, you see here. This is the normal arrangement of the upper profile that you see is the normal arrangement of the mating genes, whereas in the lower part, you see the portion of the MAT1 gene snipped away and it attaches itself to MAT2 gene. So what happens in, during meiosis is during the crossover, that little portion that you see here in the blue detaches itself and then attaches to the MAT1 gene. So this is how MAT inversion takes place in all of the ascomycetes, and this is one of the very unique features in all of the ascomycetes that we discovered. And if you look at the ascus itself, so it can be located either all four of them can be inversion plus, and four of them can be inversion minus, or two of each can be alternatively arranged, inversion plus and inversion minus. So it's a very common feature that we discovered that we later on, after we discovered these, other people discovered it in all these species that I have listed here. So the second pathosystem that I have worked on extensively is Verticillium dahliae. Verticillium dahliae is also an ascomycete, and it is a soil-borne pathogen also. It causes the vascular wilts, and it has a very broad host range, and it forms microsclerotia that you see in the bottom here, those black dots, that can remain viable in the soil for about 15 years. And in some, it can be seed-borne, and there is only in a few hosts there is host resistance available. So even before I arrived, what happened was there was a brand new disease on cauliflower. And cauliflower is a very important crop in coastal California. And is what they used to do is to put a rubber band around every cauliflower plant so that they can protect the cauliflower heads from exposing it to the sun so that it remains pristine white and they can be marketable. But what this wilt disease was doing was to collapse all these lower leaves and exposing the heads, making them unmarketable. And later on we showed that it is clearly caused by Verticillium longisporum. At that time it used to be called Verticillium dahliae. But during the course of our studies, what we found was broccoli crops were completely immune to this disease. And I wanted to see if the broccoli residue actually reduces the verticillium. So what we did was to collect the soil from very highly infested fields and sample them in small vials and put fresh or dry broccoli powder into these tubes based on what is left in a commercial field after commercial harvest. Those amounts were added to these vials, and then we incubated them at various temperatures. And as you can see here, even in the control, this one, there was absolutely no reduction, but at every temperature that we tested, the fresh broccoli was significantly better. More than 90% of all the sclerotia were reduced when the temperatures were 20 and above. So we then took it to the field and planted broccoli, incorporated residue, and both in broccoli and no broccoli plots, we planted cauliflower. And you see here, we started at the same level of the inoculum, and over 2 years, in the broccoli treatment, there was virtually zero microsclerotia in the soil, whereas when you grow only cauliflower, the pathogen increased 500-fold. So the question is, why? Why is broccoli resistant? What happens? So we studied the infection process using a GFP transformed strain, and in the upper panel is the cauliflower, and you see it goes into the vasculature very clearly. And the early colonization events in broccoli were similar, but in the cambial ring, the infection stops in the cambial ring. It doesn't go into the xylem at all. So this explained why broccoli is resistant. but it still doesn't explain how it reduces the inoculum levels in the soil when you incorporate a residue. So to assess that one, what we did was to use a metagenomic approach to identify the players that are enhanced as a result of broccoli residue in this one. You notice here, this is the control plant, and this is where the broccoli and chitin material was incorporated. Virtually healthy, completely healthy plants in these. And when we assess the players that are selectively enhanced by broccoli, you will notice this is in the bottom line. You see here the orange bars are in the control treatment, and they were completely replaced by these species that you see in the blue bars. There were totally 47 different bacterial species that were selectively enhanced as a result of incorporating broccoli into the soil. And believe it or not, every one of those species has literature, has a paper published as potential biocontrol agent. But only 3 of them have been commercialized with questionable efficacies also. So if you are a grower, do you simply rather incorporate the broccoli residue and get the benefit of all of these players together, or do you use registered biocontrol agents that have questionable efficacies? So the choice is very clear. By simply incorporating broccoli, you can eliminate many, many soil-borne pathogens. So now I will shift gears and give you a brief history of the lettuce crop and how it arrived in America and what the problem is and all that. So lettuce is the most widely consumed salad vegetable in the world. And in ancient Egypt, only the royals were allowed to eat lettuce. Ordinary commoners like you and me were not even allowed to eat lettuce. So aren't you glad that that situation has changed? And there are various types of lettuces that you see here in the picture. And in fact, Christopher Columbus introduced lettuce into North America. And since then, it had been grown in home gardens in the eastern part of the United States. But with the discovery of refrigeration in the mid-1890s, much of the lettuce production shifted westwards in California, primarily in the Salinas Valley. And now, California and Arizona account for nearly 99% of all the lettuce production in the US. And Salinas Valley alone in California accounts for 65% of all the lettuce production. So this is on the host side. On the pathogen side, the genus Verticillium was described in Germany in 1817, and the most important species of Verticillium, Verticillium dahliae, was described in 1906, also in northern Germany. And the very first report of Verticillium wilt in North America was on okra in North Carolina. But within about 15 years, the pathogen has spread throughout the United States, including California. And one of the first casualties of its arrival in California was strawberry. And strawberry, as you know, is a very, very expensive crop to grow. And you lose nearly 100% of all the plants because of verticillium. And it is to solve this problem, the fumigation with methyl bromide chloropicrin was developed in the mid-50s rather. And now you cannot use it because of its effect on ozone layer. So various other crops grown in California are also highly susceptible, and lettuce had remained immune all this time. That all changed in 1995 when an entire field came down with a new disease, and we showed that it is Verticillium wilt that causes that one. The question is, why did a host that remained immune for nearly 150 years all of a sudden become a host of Verticillium? So over the next 15 years, we researched many ideas that we typically think of as a plant pathologist. But we got some very good papers, but we were nowhere near to solving this problem. So then I went and evaluated all the changes that had happened prior to the disease developing. So before 1992, much of the lettuce in the supermarket was like this. They were either naked or wrapped heads that were sold in the supermarkets. So in the late 80s, the vegetable consumption in the US had gone down. So they conducted some consumer surveys that clearly showed that the vegetable. Can you stop talking, please? I still hear somebody talking. So what we noticed was to make it easier for the consumers to consume lettuce, they came up with this ingenious way of putting all the salad.
5 minutes.
Yes, I will complete. So complete all the salad in one bag, and this became an instant success, and it exploded in the consumer market. Now, most of the lettuce that is sold here in the US is like this. For that to happen, now the appearance, the shape, size, none of that was important. Increase in the biomass in a unit area was the most important one. And one of the field changes that happened was normally from growing 2 rows on the raised bed, they combined the 2 rows, and what was the furrow there also became part of the head. And in the same area, they increased the production by nearly 33%. And if you visit California, you see these very colorful fields where the marketer will just tell, this is the kind of mixture that we want in the salad bags. To reduce post-harvest handling, they will plant in the same proportions and harvest it and then put it in the bag. And one of the main components in these salad bags is spinach. And what is interesting about spinach is it requires between 16 to 18 hours of day length for it to flower. So the seed production is mostly concentrated in only these 4 areas. The Netherlands, Denmark produces 65% of all the seed that is consumed, that is planted in the world. And then the other players contribute a little. And they plant about 9 million seeds per hectare, and it is mowed like a lawn, and they regrow it and then mow it again so that they can harvest more tissue. And what we find out is if you examine the seed, each seed is loaded with Verticillium microsclerotia. We counted that upwards of 250 microsclerotia are located, each seed contains 250 microsclerotia and above. And imagine I told you 9 million seeds per hectare, multiply with 250, how much inoculum you are introducing with each crop of spinach. Spinach is seeded, 28 days later it is harvested. So multiple cycles of crops are grown, and you are introducing massive quantities of inoculum with each crop. And that's how it happened through some sophisticated population genetic analysis. We showed that Viswavidyalaya from spinach is ancestral to all the populations of verticillium from other crops. And if you look at the migration pattern, it is the spinach seed that introduced the inoculum into California lettuce fields, and that's what caused lettuce to become a host. And I will end the talk with this one. Outside the UC Davis library, we have these eggheads. And you can understand why these eggheads are in front of the library for encouraging the students to focus their attention on this one. When you are confronted with projects, the one that I described, where the traditional thinking of ideas